This is a parental double-stranded DNA of a bacterium called E. coli. This parental double-stranded DNA of E. coli has an origin of replication. During DNA replication, first of all, an initiator protein binds to the origin of replication of the double-stranded DNA. The initiator protein uses ATP energy to slightly unwind the DNA. This rectangular box shows one of the two replication forks in a double-stranded DNA. If we will take a closer look at this replication fork, then it will look like this. These are the two unwound DNA strands. DNA helicase having 6 subunits binds to the unwound DNA and continues unwinding. Meanwhile, DNA guidage induces negative supercoiling in front of the DNA helicase. By inducing negative supercoiling in front of the DNA helicase, DNA guidage promotes strand separation. DNA guidage is a topoisomerase. Molecules called as SSB or single-stranded DNA binding protein maintain strand separation. SSB molecules stabilize the unwound region. SSB molecules allow the separated strands to act as templates. In the next step, primase binds to the first initiating sequence on the leading strand template. Primase then synthesizes a short RNA primer. This short RNA primer is complementary to the leading strand template. In the next step, DNA polymerase III uses primer to start DNA synthesis by adding deoxyribonucleotides to its 3' end. The leading strand requires only one priming event. Because DNA synthesis is continuous in the 5' to 3' direction. This is the lagging strand template. An RNA primer is made for the lagging strand on the lagging strand template. DNA polymerase III then extends the lagging strand. In E. coli, the primase is a part of the primosome. Primosome is a protein complex. This protein complex includes both primase and the DNA helicase. For the lagging strand, DNA synthesis is discontinuous. For the lagging strand, DNA synthesis requires a series of RNA primers. DNA is synthesized at the 3' end of each RNA primer. As a result, an Okazaki fragment is produced. An Okazaki fragment grows until it meets the adjacent Okazaki fragment. For example, H2 Okazaki fragment grows until it meets the H1 Okazaki fragment. DNA polymerase I shows 5' to 3' exonuclease activity. By its 5' to 3' exonuclease activity, DNA polymerase I then removes the RNA primer. DNA polymerase I also shows polymerase activity. By its polymerase activity, DNA polymerase I places DNA on the place of RNA primer. DNA ligase links together adjacent Okazaki fragments like H1 and H2. Hereafter, DNA ligase, DNA polymerase III, DNA polymerase I, primase, DNA helicase, and DNA guidage will be working simultaneously in the neighborhood of the replication fork. Please like, subscribe, and share.